Good afternoon, BSc second year students. Now we discuss the unit paper number 7th, unit 2nd, that is the enzymes. All of you know that is the enzyme is related to the chapter that is the proteins. I have given you information about the proteins in unit 1st. What is meant by proteins? What is the structure of proteins, what is the classification of proteins, and properties of proteins, physical and chemical properties. Basically, that is the enzymes. In unit 2nd, that is the enzymes. Enzymes is very important article. These enzymes discovered by the Cuny scientist in 1878. Enzyme first discovered from the Greek words by yeast. Yeast is a cell. Removal of the enzymes in all the living organisms, and proteins are soluble and colloidal cell exist without enzymes. Basically, that is the enzymes and proteins are very important, compact. It is the base of biocatalyst. So the Cuny scientist introduced the 1878 and studied the enzymology. What is meant by the enzymology? Enzymology is nothing but the study of enzymes. That is known as the enzymology. And all the enzymes are located in the cell. And what is the structure of cell you studied? That is the plant cell and animal cells. Yes, you know that is the cell, cytoplasm, mitochondria, tissues of the body fluids. Yes, that is the information about the cell. Cell is nothing but the structural and functional unit of the living organism. Basic structural and functional unit of the living organisms. All of you know that is the simple definition of the enzymes. Yes, enzymes are the proteins. Mind well, enzymes are the proteins and are biocatalyst, biocatalyst of the biochemical reaction in the living organisms. Student, one thing remember it, enzyme contain proteins, and I have also described and given hint about the enzymes. All enzymes are proteins, but all proteins are not enzymes. This thing remember, mind well. Here may be that is the enzymes, there may be that is the proteins. So all the enzymes are proteins, but all proteins are not enzymes. Enzymes are the mind well the simple definition. Enzymes are the proteins. Biocatalyst. What is biocatalyst? Is nothing but bio means living. Before the bio, you must know that is a bi. Bi means two. That is a plant and animal. Already I have given the information about the plant cells and animal cells. Yes, plants and animals, these two. Enzymes are present inside the plant cells as well as animal cells by and living both the characteristics the enzymes are present, plant cells and animal cells. So it is known as the biocatalyst. Protein is very important, mind well. And biocatalyst, and it is the protein take reactions in the biochemical reactions. Bio means living, chemical reactions of the living cells, which may be that is the living cells where the plants and animal cells, both the. So given the word biochemical reactions in the living cells. Yes. What is the nomenclature? What is the nomenclature of the enzymes? Now we discuss about the nature. What is the nature of proteins? In the nomenclature, you must be know that is the name of the enzymes is called nomenclature of enzymes. Nomenclature of enzymes. Enzymes are based on the substrate. Enzymes are based on the substrate. Enzymes are based on the reactions. Enzymes are based on the synthesis, and enzymes are based on the chemical reactions. These are the basic things about the enzymes. Yes, when you studied that is the substrate, 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 some reactions, synthesis is nothing but the form, and some chemical reactions, those present from the enzymes. All these things are present in the nomenclature based on the substrate, isn't it? What is meant by the substrate? Substrate is an enzyme act, may be named as the suffix ase. Yes, as and given the substrate, name of the substrate. substrate. For examples, already I have given the information about the enzymes. Enzymes contain the proteins, and protein contain amino acids. Yes, and amino acids contain the carbohydrates, and carbohydrate contain the major, that is the in the classification of proteins, that is the carbohydrates. And best examples which may be that is the glucose and fructose. Same molecular formula but different structural formula. Molecular formula C6H12O6. Yes, and different structural formula in glucose contain aldehyde group, CHO group, and fructose contain ketonic group, that is known as the hydroxylic group or ketonic group. That is the difference between the glucose and fructose, and this best example of carbohydrates. When you read the enzymes act as a carbohydrates in the nomenclature and named as the carbohydrates. Yes. Second, enzymes act as a protein and name proteinase. ASE suffix I have given last 3 digits. RATES, carbohydrates and protease, TASE, protease. And second group, which may be that is the lipids. What is the acting of lipids? Acting of lipids and named as the lipases. LIPASE, lipase, lipases or lipases. Yes, enzyme act on the nucleic acid and that is named as nucleases. NUCLEASES, nucleases. Lipids, lipases, LIPASES, lipases. Proteinases, PROTEINASES, proteinases. Yes. And firstly I have given that is the carbohydrates. And these types that is the basically the enzymes based on the substrate reactions, synthesis and chemicals, and that is the simple nomenclature given in the carbohydrates, proteins, lipids, yes, and some chemical reactions. And some important characteristics also in the nomenclature about the carbohydrates, about the proteins and about the lipids. It is a basically all these components are present. All these components are basically present inside the inside the enzymes. The enzymes acting as a maltose. Enzyme acting as a maltose and named as maltose. Maltose is basically carbohydrates. When you studied in the maltose, that is the basically carbohydrates and its classification, basic classification and classification and structure also describe the maltose. Maltose, lactose, urose, sucrose, and these types. Next characteristics in the nomenclature, the enzymes acting as a lactose and named as a lactase, LACTASE, last TASE. Maltose named as acting and named as a maltase, MALTASE. Yes, lactose acting, lactose named as a lactase. Enzymes acting on urea, that is urease, UREASE. These are the important things in the synthesis of the carbohydrates, proteins and lipids. I have given that is in the nomenclature enzymes based on the reactions. Yes, and that is the suffix, that is last word change in the reactions. Last word change. I have given that is the some hydrolysis. Yes, enzyme acting as a hydrolysis and named as a hydrolase, HYDROLASE, last this suffix hydrolysis, HYDROLYSIS, yes, and named hydrolase. Spelling, students mind well, that is the hydrolysis. Hydrolysis is nothing but the addition of water. Yes, that is known as the hydrolysis. Acting, enzymes acting hydrolysis and named hydrolase. You know that is the next one. Enzymes catalyze the oxidation, and it is named as the reductase. Reductase. What is mean by oxidation and what is mean by the reduction? Oxidase, reductase, last ASE. That is the change in the last words when the enzymes take the reactions in the plant cells and animal cells to change the factors last three words. Can you study it in detail? Isn't it? Enzymes catalyze the dehydrogenation. Dehydrogenation, removal of hydrogen. Dehydrogenation. I named dehydrogenase. Mind well this spelling. Enzymes catalyzing dehydrogenation. DEHYDRATION. Dehydrogenation. I named as dehydrogenase. 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 Yes. These types. Next reaction in the enzymes. Enzymes catalyzing phosphorylation. 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 Yes, and named as phosphorylase. 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 Yes. These types, that is the reactions. In the first I have given hydrolysis, named hydrolysis, oxidase, reduction, reductase, dehydrogenase, yes, phosphorylation, phosphorylase, LASE, last suffix. Enzyme catalyzing as isomerization, ISOMerization, isomerization. Iso means same, merization. I have given these characteristics and described already. Where? In the proteins, carbohydrates or lipids? No, I have described in detail, that is the isomerization in the carbohydrates. Now just describe the same molecular formula but different structural formulae. Yes, C6H12O6, name glucose or fructose. That is the same molecular formula, that is no isomerization, isomerize. And catalyzing named as isomers, ISOMERASE, isomerase. Yes, that is the in nomenclature the reactions. Is that in, students? I have given in the nature of the enzymes based on the substrate and reactions. How many types of the reactions present inside the enzymes? The number of the reactions present inside the enzymes. Mind well, it is a biocatalyst. Enzyme is very important characteristics, biocatalyst of the living organisms, cell of living organisms, to form the substrate and reactions, are based on the substrate and reactions. Which is the based substrate and reactions, and what is the types? Enzymes remove carbon dioxide from pyruvic acid, and named as pyruvic decarboxylase. Pyruvic decarboxylase. Yes. What is the, I have given that is the reactions and substrate and reactions in the enzymes. And these types, my dear students, enzymes based on the synthesis and name change the suffix means last words, ASE, synthesis, to the substrate, to the synthesized. What is the enzyme synthesizing citric acid? Citric acid, and it named as a citric synthetase. Named as citric acid. What is mean that citric acid to citric synthetase? SYNTHATHAETASE, last word. Enzyme synthesis citric acid and named as a citric acid synthetase. Yes, SYNTHETASE, synthetase. These types, some changes in the enzymes from the synthesis, and to change last words, that is ASE. I have given that is the based on the some examples which are related to the proteins. What is the amines? Protein contain the amines, and some protein to form the synthesis. Pepsin is the basic part of the proteins. Yes, pepsin, trypsin. Enzymes named as and discover these. Some components are present inside the pepsin, trypsin, petalin. These are the best name which may be that is related to the proteins. Mind well, students, these are the basic things. Enzymes, when you read carefully at the unit first, its classification of the carbohydrates, structure of carbohydrates, and some properties of carbohydrates. Next one, that is the proteins. Enzymes base protein. Already I have given the proteins. Proteins are the base of enzymes, and all proteins are the enzymes, man. And but proteins are, all enzymes are protein, but all proteins are not enzymes. Man, that is the slogan. Simple. Yes. You read carefully, that is the structure of proteins, classification of proteins, classification based on the chemical reactions, classification based on the other factors, substrate, synthetic, these types. Yes, and properties of the proteins, which may be that is the physical properties and chemical properties. At that time you will know. And third part important, that is the lipids. Lipid synthesis, that is the lipids. Basic things, and these basic things are included under the in the unit second unit, that is the enzymes. I have given that is the definition of what is meant by enzymes. Definition of enzymes. Yes. Discover of enzymes. Basic things about the enzymes which are related to carbohydrates, proteins and lipids. And last verse, which is the nature of the enzymes, and important article, that is the nomenclature. Yes, today we discuss the nomenclature based on the synthesis, reactions and substrate. These three things I described just. Yes, these three things is very important. Now we stop here. Remaining, you read carefully and listen this video and give comments if you have any difficulties or any problems in the talking. If you have any problem, to contact me and give feedback. I will recover these points. If some points are repeated, some points are again repeated, or some point not described, you tell me, and with maybe that is I will give a lecture, and that is the some problems about the talking or net problem. You comments me, I will recover these points. Thank you. Let it go.